Hello. In this video, I'm going to describe the process of calcium homeostasis. To clarify, we as humans maintain the levels of calcium in our blood at about 8.5 to 10 milligrams per deciliter. Of course, at all times, we are ingesting calcium or we are depositing it into bones. We are excreting it via our urine, via our feces. And so there are a lot of different ways that calcium levels can be adjusted. It is really important to maintain calcium levels at this particular level. So we'll say about 9 milligrams per deciliter. Because our muscles utilize calcium and, of course, gradients of calcium across cell membranes. Our neurons use calcium, and so many other cells in our body require a particular concentration gradient of calcium between inside the cell versus outside the cell. And so it's very important to maintain calcium within this narrow range. The first scenario I'm going to talk about is a hypercalcemia scenario. Hyper means too much or above this normal range. Calc for calcium, and emia means in the blood. So hypercalcemia. So essentially you just had a big glass of milk. Okay, so now your calcium levels within your blood are a little bit higher than they otherwise should be. And so the organ that is responsible for monitoring this hypercalcemic condition is the thyroid gland. What I have for you up here is an image of the trachea. So your windpipe, and a little kind of like bow tie-like shaped organ. Over the, or anterior to the trachea, is the thyroid gland. Here we are looking at the anterior thyroid gland. In the next part of this video, we'll talk about the posterior thyroid gland as well. And so again, this right here happens to be this right here, right in your throat. Okay, so if we zoom in to the anterior thyroid gland, we see that most of the tissue is made up of follicles. Now, these follicles are going to have another function. They are responsible for producing thyroid hormones. See this other video if you want to hear more about that. But the cells that are responsible for monitoring calcium are called parafollicular. Right? So they're adjacent to the follicles. They're also sometimes called C cells. Okay? And so essentially, as blood flows past these C cells, the calcium is going to bind to calcium receptors or calcium channels. And essentially, this is going to trigger a chain reaction within the parafollicular cells to exocytose, to release calcitonin into the blood. And I like to remember calcitonin is released because there is a ton of calcium, again, emia, in the blood. Yes, calcitonin is released when there's a ton of calcium. So what does calcitonin do, here abbreviated CT? Well, of course, it's going to circulate throughout the entire body within the blood, and it's only going to bind to receptors on target tissues. And so if there is a calcitonin receptor on a tissue, it is going to induce some pre-programmed response. The first really primary target tissue of calcitonin is on the bones. Okay, so essentially, if you have too much calcium in the blood, we don't want to just, get rid of all of that, right? You had that glass of milk and you want to make sure that you can save that calcium to strengthen your bones. And so the way that we strengthen our bones is calcitonin is going to bind to calcitonin receptors on particularly one type of bone cell. This bone cell is called an osteoclast. Now, just as a very brief background here, osteoclasts are responsible for essentially breaking down the bone. Okay, so kind of burrowing tunnels into the bone and liberating all of the different components within the bone. Okay, so osteoclasts break down the bone, as opposed to osteoblasts. Right? Blast here indicates a building cell. And so osteoblasts are responsible for taking calcium and other components out of the blood and secreting, so taking calcium out of blood, as well as phosphate, right? And creating proteins such as collagen and proteoglycans. And there, of course, this story is a lot more complicated than this, but essentially osteoblasts secrete this organic component of the bone called osteoid, containing these two components, and they essentially increase the calcium concentration within the localized area. Together, the calcium and phosphates are going to spontaneously form crystals, right, called hydroxyapatite, sorry about that, on this existing protein organic component of the bone, right? So osteoblasts are building the bone, and by building the bone, they remove calcium from the blood. Okay? And there's kind of this balance between osteoclasts and osteoblasts. These guys are building at the same exact time osteoclasts are breaking down the bone. And this is a good thing. It might surprise you to hear that your bones are constantly being broken down. But in reality, this helps us to replace our bone tissue so that we are constantly having new, healthier bone tissue. And so let's get back to calcitonin. Calcitonin binds to its receptors on the osteoclasts and essentially inhibits them. And so it's inhibiting the breaking down part of this process, and the osteoblasts are going to proceed on as usual. So they are going to continue to take calcium out of the blood, deposit it into the bones, and therefore strengthen the existing bone tissue. So in this way, high calcium bound to receptors on C cells of the thyroid gland induces the release of calcitonin, which inhibits osteoclast activity and allows osteoblast activity to essentially outweigh the breaking down components. Okay, so that's what happens when you have too much calcium in the blood. If you have too little calcium in the blood, so hypocalcemia, too little calciumemia in the blood. Too low of calcium is detected not in the thyroid gland, but in the parathyroid glands. These are 4 condensed regions on the posterior side of the thyroid gland. Okay, so on the posterior side only. There are a couple different types of cells within the parathyroid glands, but the ones that we are focused on in this video are the chief cells. The chief cells have also receptors and channels for calcium. Okay. If there is a lot of calcium in the blood, essentially this high calcium scenario inhibits the exocytosis of a hormone. Okay, so just a little bit of background here. At all times, these chief cells are transcribing a hormone, right? So transcribing a gene within the genome. That mRNA, right, is translated out in the cytoplasm. This mRNA produces a protein sequence that is about 115 amino acids long. We call this pre-pro PTH, parathyroid hormone, or parathormone, depending on what source you're looking at. This pre-pro PTH is going to be processed by the rough endoplasmic reticulum. In the process of this, of the RAR's function, it is going to cleave off 25 of these original amino acids, leaving a 90 amino acid long pro-PTH. But of course, the story is not over. Pro-PTH is sent to the Golgi apparatus, and the Golgi apparatus is going to cleave off the final 6 amino acids. And finally, we have this parathyroid hormone. It is 84 amino acids long, but in reality, only 34 of these are active in the process of maintaining calcium homeostasis. So after all of this transcription, translation, processing, the Golgi apparatus is going to send vesicles to the surface of the cell, and these vesicles are chock full of parathyroid hormone. They just kind of sit there and wait. Essentially, as I was saying, high calcium is going to inhibit the release of these hormones. They're just going to be waiting for a situation where calcium drops. And this is important because we need to be ready to go. We really don't want calcium to drop any lower than it should be, because it can have detrimental effects on neuron function as well as muscle function. Okay, so the body is ready to release PTH, again, parathyroid hormone, also called parathormone. However, when calcium is reduced, calcium is going to bind more infrequently, right? So not as often, to these same receptors or channels. And so that is going to exert less inhibition. Right? This has lots of inhibition, and so PTH is held within the vesicles with less inhibition. There's going to be exocytosis. So here we see this release of PTH into the blood. And just like we saw with calcitonin, PTH is going to circulate throughout the entire body. This time, PTH is going to be active on a series of different organs. Okay, so before we can understand what exactly PTH is doing, let's rewind a little bit and talk about what is happening at the skin when you go out in the sun. Okay, so what we can see here is the epidermis, right, the surface of the skin. When you go out into the sun, UV rays are going to produce a conformational change of this molecule right here, 7-dehydrocholesterol. Okay, so this is a steroid that is constantly within your skin. UV rays are going to change this molecule, and we call it cholecalciferol. Okay, we also have another name for it, which might be more familiar to you, and that is vitamin D3. Okay. Just a side note, vitamin D is kind of a blanket term for a few different types of molecules, all in this same family. Okay, so the sun produced cholecalciferol. Cholecalciferol enters into the blood. Okay. Ultimately, again, circulating all throughout the body in the bloodstream. Ultimately, it is going to go into the liver. And the liver has a lot of different enzymes, lots of different functions, but here we care about a mitochondrial enzyme called 25-hydroxylase. When cholecalciferol passes through the liver, this enzyme acts on cholecalciferol, and it's going to change it yet again. Right? In general, it is cleaving off hydroxyl molecules. So the new molecule, right, ultimately came from the skin, through the blood, to the liver. This molecule is also just one more step in the process. And so it is going to be dumped back into the blood, again, circulating all throughout the body. When the kidney filters the blood, that's the kidney right here, it is going to produce the active form of vitamin D. Okay, so 25-HC is going to go into the blood, and the kidney is going to convert it. Okay, so if we zoom in to this portion of the kidney, we can take a look at one single nephron. In the nephron, blood is filtered over here in the renal corpuscle. Okay, and so the filtrate from the blood, including this molecule up here, is going to pass through and ultimately can be ejected or excreted as urine. But in the presence of PTH, parathyroid hormone, this molecule is converted by yet another enzyme to the active form of vitamin D. And of course, why name something once if you can't name it 2 or 3 times? Active vitamin D is also called calcitriol. And I like to remember calcitriol tries to increase calcium. All right, again, the problem is we don't have enough. So calcitriol tries to increase calcium. More on that in just a moment. And we could also call it 1,25-DHC. Okay, so again, in the presence of PTH, this molecule is converted into the active vitamin D, which tries to increase calcium. Otherwise, this molecule is going to continue on its journey through the nephron and be excreted as urine. Okay. Before we leave the kidney, let me also talk about another thing that PTH is responsible for. PTH also binds to receptors on the distal convoluted tubule, and through a series of pre-programmed events, PTH leads to the insertion of more calcium channels in the membrane of the distal convoluted tubule, therefore increasing how much calcium is going to be reabsorbed into the blood. Okay, so if any calcium was left over in the filtrate, it is instead going to be reabsorbed back into the blood. Right, so this is the first time in this story that we see calcium increasing in the blood. Again, the problem here is that there's too little calcium. Well, how do we fix the problem? We increase calcium in the blood. Another way of increasing calcium is calcitriol, that is active vitamin D. again, circulates throughout the body in the blood and ends up in the intestinal blood. Okay, so here we can see calcitriol binding to a calcitriol receptor on the membrane of the intestines. In response to calcitriol binding to its receptor, we are going to increase how much calcium binding protein and calcium ATPase, right? So essentially locking onto calcium that was in your food, okay, and pumping it into the intestinal lining cells, the mucosa, and letting it diffuse into the blood. Okay, so far PTH has actively taken more calcium out of the soon-to-be urine, essentially, and it has actively pulled more calcium from the food into your blood. Now, of course, if calcitriol, that is PTH, was not released, then this calcium would instead end up in the feces. Okay, but in this situation where PTH has been released, there's going to be less calcium in the urine because it's now in the blood, and there's going to be less calcium in the feces, again, because now it's in the blood. Okay. One more piece to this puzzle. PTH has many widespread effects throughout the entire body, which should indicate to you that the body thinks it's really important to not let this scenario happen. You know, we have a lot of fail safes. We don't pee any out, we don't release any as feces, and we can actually dig into our reserves within the bones. Okay, so again, PTH is circulating throughout the body within the blood, and PTH binds to, in this case, osteoblasts. So here we have PTH binding to a PTH receptor 1 expressed on the surface of the osteoblasts. This ligand binding to its receptor is going to lead to the release of a molecule called rank. Okay, so just a chemical signal, which is going to bind to neighboring osteoclast cells. Via this indirect interaction, osteoclasts are really going to increase their activity. Okay, so over here, calcitonin decreased their activity so that calcium wasn't liberated. Now osteoclasts are going to do what they do best. They're going to spit out enzymes, they're going to spit out acid, and essentially burrow tunnels into the bone. As the osteoclasts break down the bone, everything that was in the bone, that is proteoglycans, collagen, phosphate, and calcium, is going to be liberated and deposited back into the bone. So here we can see the bones actually serve as a calcium store. Okay, so this reservoir of calcium is utilized to increase calcium levels within the blood. Furthermore, not only does PTH directly increase bone resorption, that is liberation of calcium, also calcitriol, that is the vitamin D, right, made by the skin, modified by the liver, and then activated by the kidney. Calcitriol binds to calcitriol receptors on osteogenic cells within the periosteum. Okay, so here, what we see is a membrane that surrounds most of the bone. Right? As part of this layer, there are osteogenic cells, which essentially means stem cells. And these stem cells can essentially be triggered to essentially grow up and become either osteoblasts or osteoclasts or a couple other things. And so calcitriol is going to stimulate these stem cells to differentiate, right, grow up to be or start expressing different genes to be an osteoblast and osteoclasts. And so essentially, by doing this, we have more cells that have PTH receptors, and we also have more osteoclasts that can increase the resorption of the bone. Okay, so once again, this is the process of calcium homeostasis. The thyroid gland secretes calcitonin to decrease bone resorption when there is a lot of calcium in the blood. When there is not enough calcium in the blood, the chief cells of the parathyroid glands, which happen to be on the posterior side of the thyroid gland, this low calcium level is going to trigger the chief cells to release PTH, parathyroid hormone, into the blood. PTH is going to increase the calcitriol or active vitamin D levels within the blood. It is going to increase calcium reabsorption from the kidney. It is going to increase calcium absorption from the intestines into the blood, and it is going to increase bone breakdown, right? And therefore liberation of calcium to once again increase calcium in the blood. Thank you so much for watching. I hope that this helped you to understand calcium homeostasis.